Assalamu alaikum, dear students. This is me, Atiq Ahmad. Today we are going to study the topic Meiosis 1. Before proceeding towards Meiosis 1, we will have an overview of the previous lecture. In the previous lecture, we have started Meiosis. Meiosis is the type of cell division in which one cell is divided into four cells. And in this process, the number of chromosomes in the daughter cells is reduced to half as compared to the parent cell. The meiosis process takes place in the six cells, and the sperms and eggs are produced through the process of meiosis. The interphase or preparatory phase, or in other words the resting stage, which we have already studied in the mitosis, that is similar in mitosis as well as in meiosis. That is the preparatory phase, and in the interphase the synthesis of DNA and various enzymes takes place. The organelles are also duplicated. The interphase consists of three subphases: the G1 phase, the S phase, and the G2 phase. So the interphase is similar in mitosis as well as in meiosis. The meiosis is broadly divided into two major stages: the meiosis 1 and the meiosis 2. So after the interphase, the cell will enter into the meiosis 1 first, and then it will enter into the second major stage, the meiosis 2. Today we are going to study the meiosis 1. Meiosis 1 consists of the following stages: prophase 1, metaphase 1, anaphase 1, and telophase 1. Today we will study the prophase 1 of meiosis 1 in detail. This is the most important phase of the meiosis 1. It is the longest phase in meiosis, and it accounts for 90% of the total time spent in meiosis. During this stage, chromatin condenses into chromosomes and become visible under the microscope. This is similar to mitosis. Just like in the prophase of mitosis, the chromatin condenses into chromosomes. Same is here in the meiosis, that in prophase 1, the chromatin condenses into chromosomes, and those chromosomes are prominent and are visible under the microscope. The homologous chromosomes, those chromosomes which are similar in shape and in size, they line up with each other and form pairs by a process called synapsis. Synapsis is the process in which the homologous chromosomes, those chromosomes which are similar in shape and in size, they pair up with each other, and they form pairs. For example, if we take the example of human cells, so in human cells there are 46 chromosomes. So whenever the meiosis started, so the number of chromosomes is doubled in the interphase, and when the meiosis 1 starts, so then the chromosomes are condensed, the chromatin is condensed into the chromosomes, and they start pairing up. So pairs are formed. This process of pairing is known as synapsis. Diagrammatically, it is shown here. This is prophase 1. Here it is shown that the homologous chromosomes, these two are homologous chromosomes, the blue and the red one. So they line up with each other and form this pair. Similarly, this is another pair, this is another pair, so there will be several pairs. So this process of pairing, this is known as synapses. In another diagram, it is shown here. Homologous chromosomes are aligned, or in other words, they are line up with each other, and this process is known as synapses. These two are homologous chromosomes and they are paired up with each other. Each pair of homologous chromosome is called a bivalent. Each bivalent has 4 chromatids, so it may also be called as tetrad. The two non-sister chromatids of the homologous chromosome join each other at certain points along their length. These points of attachment are called chiasmata. Singular is chiasma. Now, whenever the homologous chromosomes pair up through the process of synapses, so a pair is formed. That pair, just like it is shown here, or in another diagram it is shown here. Now this is one pair. It consists of 2 chromosomes. These 2 chromosomes, when paired up, these are known as bivalents. This is one bivalent. It consists of 2 chromosomes. Bi means 2 in Greek. So it is bivalent. It consists of 2 chromosomes. It is also known as tetrad. Tetra is for 4. In Greek, tetra means 4. It is also known as tetrad on the basis of chromatids, because this pair consists of 4 chromatids. 1 chromatid, 2nd chromatid, 3rd chromatid, and 4th chromatid. So whenever the synapsis process takes place, in the synapses the chromosome pair up and pairs are formed. The homologous chromosomes form pairs. Each pair consists of 4 chromatids, so it is known as tetrad. It is also known as bivalent. Each pair is also known as bivalent because it consists of 2 chromosomes. So it is bivalent or tetrad because it consists of 2 chromosomes and 4 chromatids. Now, next point is that they will attach with each other at certain points, which is known as chiasma. So this is the chiasma. Chiasma is the point at which the 2 homologous chromosomes make contact with each other. These 2 are homologous chromosomes. They form this pair during the synapses, and then certain parts of these chromosomes, they make contact with each other. This is the point of contact. This point of contact is known as chiasma. Its plural is chiasmata. So each pair of homologous chromosomes is called a bivalent because it consists of 2 chromosomes. Each bivalent has 4 chromatids, so it may also be called as tetrad. It may also be called as tetrad because of 4 chromatids. The 2 non-sister chromatids of homologous chromosomes join each other at certain points along their length. These points of attachment are called chiasmata, singular chiasma. We have already seen in the diagram. In the next stage, the non-sister chromatids of homologous chromosomes exchange their segment parts, and the phenomenon is known as crossing over. Now it is shown here in this diagram that the non-sister chromatids, this is one non-sister chromatid, this is another non-sister chromatid, they have attached at this point the chiasmata, and at this point they exchange their parts with each other. So this part will move towards this and this part will move toward this. So these non-sister chromatids, why these are called non-sister chromatids? Because this and this chromatid, these belong to one chromosome, so they are sister chromatids. But this one and this one, the red one and the blue one, they are non-sister chromatids because they belong to two separate chromosomes. So during the process of crossing over, the two non-sister chromatids, this is one sister chromatid, this is another one, they will exchange their parts with each other, and this process of exchange of parts of chromosomes with each other, this process is known as crossing over. Here it is shown that the parts has already exchanged. This red one is attached with the blue one, and the blue part is now attached with the red chromatid. So this exchange of parts, this is known as crossing over. In the next step, the exchange of segments result in the recombination of genetic information. Now, whenever the parts are exchanged by the non-sister chromatids of the chromosomes, so this process result in the recombination of genetic information, because this is one sister chromatid, this is another sister chromatids. Whenever they exchange their parts, suppose this part moves here and that part move here, so the genetic makeup will be changed because the genetic sequence is now changed here. The original part is removed and another part is attached here with this chromatid. So this is known as genetic recombination, because the genetic information is exchanged through those parts which are exchanged between the two non-sister chromatids. After crossing over, each pair of homologous chromosomes remain as bivalent. As after the crossing over, the homologous chromosomes will not separate from each other, rather they will remain in the same form as a bivalent, as a pair of two chromosomes. Now, there are certain changes which occur in the prophase of mitosis, as well as those are similar here in the prophase of meiosis, that the nucleoli will disappear and the nuclear envelope will disintegrate. It will be broken down, the nuclear envelope or the nuclear membrane. The centriole which were already duplicated during interphase, they will migrate to the two poles and will form spindle fibers. These changes are similar to that of prophase of mitosis. So to sum up, during the prophase 1, the various changes which are occurs, we will have a summary of that. So in the first step, the synapses will occur. The chromosome will come close to each other, and the homologous chromosomes, the similar chromosomes which are similar in shape and size, they will form pairs. Secondly, they will attach with each other at certain points, and that point of contact, that is known as chiasmata. The third change which occurs during the prophase 1, that is the crossing over process, that at the point of contact, at the chiasmata point, the non-sister chromatids of the two homologous chromosomes, they will exchange parts with each other. And the next changes which occur here, that is that the nucleoli will disappear, the nuclear envelope will break down and will be removed, and the centrioles will migrate to the opposite poles, and they will form spindle fibers. This is all about today's lecture. Inshallah, see you next time with new lecture. Take care. Allah Hafiz.